Good morning, one and all. Thanks to the organizers for giving an opportunity to talk at this international forum. My talk is basically an extension. I would say Professor Lee has made a background. He has made a nice background for computational chemistry, bioinformatics, and chemoinformatics. I'm extending from small molecule drug discovery into peptide design. Many drugs like insulin and certain other drugs, they are all peptides in nature, and designing peptides is a complex task. The first slide I look upon is problem and need. Now, designing a peptide in an experimental lab, is governed by multiple parameters. First is you need to know the 20 amino acids which you wish to incorporate, and the nature of that amino acid that you would be requiring. Again, the conformation that structure or that peptide can assume, and the length of the peptide. Like you can have small dipeptides like ACE inhibitor dipeptides. You could have 21 amino acid insulin peptides. You could have bradykinin peptides, which are 5 amino acids. You could have major histomobility complex 9 amino acids. So like if you have to make a data set for experimental evaluation just for 2 amino acids, then 2 amino acids combination will be a large data set. You'll have almost 400 peptides, dipeptides, when it is taken in combination, like 2 to the, permutation combination. So synthesis is a long process, getting the amino acids and procuring them, synthesizing, and then testing them. Amino acid scan is another experimental technique wherein if you know a particular peptide active against a particular target, you can keep on constantly changing that amino acid one by one at a position, and then you can perform activity, and you know whether it's an active or a deactive. Specifically I'm talking about the ALA scan. Again, that's time consuming. I wouldn't say that experimentals is not required. Experimentals is required because experimental forms the basis of bioinformatics and chemoinformatics. It is the rich source of data from where you can go to the next level. Your computational techniques will help to simplify certain processes to achieve targets a bit faster. The in silico methods that can be used is biomolecular dynamic simulations, wherein you track the trajectories of the compounds. You can do QSAR, basically that is quantitative structure activity relationship methods. Basically what you do is you know the structure, you know the fingerprint of the structure, you know the physicochemical properties like the solubility, the lipophilicity, the shape of the molecule, and that can be quantified in terms of numbers, and then you link those numbers to the biological activity in terms of using a statistical program like maybe partial least squares, or you could use genetic functions, and you can devise a model. A simplest model that we all must have learned is the straight line, that is y is equal to mx plus c. That's the straight line equation. So QSAR looks an extension of those straight line equations. Now QSAR has been used for a long time for small molecules, small organic molecules, and peptide QSAR is in its infancy stage, wherein we're trying to adapt QSAR for peptides. It's a cumbersome task, but yes, not difficult. Now the idea here is to develop predictable models. You can use models which can predict activity for certain position of the amino acid in a particular peptide sequence. Now again here the restriction is you cannot do a cross prediction. What I mean is, if you have developed peptides or synthesized peptides against a particular target A, you can use that model which is developed for only that target A. You cannot use it for target B. So you will have to redesign the model. So it's not a universal model. You'll have to redesign it as per the target requirement. Then the basic QSAR was founded by Corwin Hansch. He is the father of QSAR. So the Hansch method basically uses descriptors, as I talked about, solubility, physical parameters, electrostatic parameters, And another method referred to as the free Wilson method uses basically binary digits of zeros and ones. So it indicates whether a group or a structure is present or absent, and based on that, a model can be devised. So using these techniques, I have tried to move into the peptide QSAR world and coined a few methods which are basically called as descriptor-based peptide QSAR, binary peptide QSAR. Then another method which takes into consideration 3D alignment. And alignment is a stacking of certain structures and looking at comparing their similarities. So that method is referred to as HOMOSAR. That is basically blends two methods, that is homology modeling and QSAR. And another method called as ensemble QSAR, which the word ensemble indicates that when a drug binds to a receptor, it is not in one single conformation but a cluster of conformation. So it's in a dynamic process. It's not a one static structure that binds. So basically we are looking at an ensemble, multiple conformations contributing activity. So coming to descriptor-based QSAR, the objective is simple, just develop a QSAR model based on the physicochemical properties. As I said, enlisted physicochemical properties like solubility, polar surface area, hydrogen bonding ability, lipophilicity. Now here, every amino acid, all of the 20 amino acids, natural amino acids, can be described using physicochemical properties, either derived from literature, because there's a lot of literature available wherein these physicochemical properties have been experimentally determined for these 20 amino acids. And again, if some of these properties can be calculated using theoretical methods, maybe ab initio calculations. So basically using these methods, we develop a model using genetic functions and then try to correlate with them with the biological activity. So this is how a descriptor-based model would look for. This is for a data set called as MHC class of peptides. These are non-amer or 9 amino acid peptides. There's a huge database from Basel, Switzerland by Flower Group. So they are quite active in this MHC class, that is major histocompatibility complex. So the model this is a representative model developed. Okay, this work has been published in QSAR and CombiScience way back. The model basically looks at the biological activity be correlated to multiple parameters here, and these parameters may be looked upon as like this is lipole moment, then there is accessible surface area, then certain dipole moment. Okay, so multiple parameters which are either theoretically determined or experimentally obtained for amino acids can be correlated. For example, this is a parameter that is MS WIM parameter 3 for position 1. So it is for position 1 amino acid. So if it's a non-amer peptide, it will look at multiple positions here. Some of only those positions we can see that there is a contribution or there is a coefficient will tell whether there is a contribution, positive contribution or a negative contribution. Then this is basically the test set on which the model has been devised. Now when it comes to in a graphical representation of how this model would look, this is basically a histogram indicating the properties which on increasing the values would provide a positive contribution to the biological activity of that particular peptide, or these would be the negative contributing properties. So if you make a negative change in this property, then definitely the biological activity of that particular peptide would increase. So if it's like here, if you try to decrease the dipole moment of the 7th amino acid, okay, for example, then you are bound to increase the biological activity as per the model. Next, which talks about the binary QSAR. As I said, the binary indicates in the computer language 1, 0 or 1, present or absent. So here again the model, it basically looks whether the amino acid is present at that position or no, and then it looks at vicinal position, that means what are the neighboring amino acids. So this is how a model would look. Again, the biological activity being correlated to the amino acid. So here, if you look at it, it talks about whether you require that amino acid or not at the particular position. So here it is aspartic acid, here it is alanine, here it is arginine, here it is threonine, valine, and what is the contribution of each of these amino acids at that particular position. Say for example valine, you have got 0.43 contribution towards the 9th position. So it indicates valine is an essential residue at the 9th position of this particular peptide. Again, same here is a histogram representing now. Earlier it was descriptors, that was the chemical properties of those amino acids. Here it is the amino acid itself. So whether you need an amino acid or you don't need an amino acid. So for example, at position 2, a leucine residue is accepted. At position 1, arginine or a tyrosine is accepted, but not to that great extent. At position 7, a glutamine would be unacceptable. So if you try to make a peptide with glutamine at the 7th residue, this would not be accepted. The biological activity would fall down. So this is basically a representation. It takes into consideration mostly all the 9 amino acid positions. Again, this is taking into consideration the individual amino acid position, because we see that any position is always affected by the environment. So if I'm sitting in one position, the person sitting next to me will definitely affect my behavior at that moment. So in this case, it also takes into consideration as this position. Can you see that looks at glycine and threonine. So glycine at position 1 and threonine at position 2. So if you have this combination in the peptide sequence, it would affect the activity, it would decrease the activity. Or else if you have a phenylalanine at a 5th and the 6th position simultaneously, you are bound to have a greater activity. Okay, so this is wherein it looks at the position, self position, as well as the position of the next neighbor. Now again, these methods what have been developed is based on the data set that is available with biological activity on the web world. The third method looks something called as homology QSAR, or that's Homo QSAR or HOMO-SAR. When we look at binding of certain inhibitors to the protein site, the problem that encountered is what is the binding conformation. Okay, if it's a rigid structure, then it would bind in a particular conformation, but if it's a flexible ligand, it could adopt multiple conformations. And it is in the absence of the receptor, it is very difficult to determine what would be the binding conformation. And now if you have a peptide, a small molecule would have say for example 2 to 3 rotatable bonds. Okay, so a small molecule, whereas peptide, you have got phi-psi angles, so, and which can rotate over multiple conformations. So if you just have a dipeptide with if you have a dipeptide, one dipeptide, and if you rotate the phi-psi over, say the number of combinations you could have is 360 divided by the number of conformations, okay, based on the rotating degree of the, so you could have multiple conformations. So exactly identifying the conformations from a 3-dimensional structure is difficult. So herein we adopt what is called as the 2D alignment. The 2D alignment is basically a sequence alignment of the peptides, where very widely used software in this case is Cluster W, Cluster X, wherein sequences are aligned with one another. So using I could better explain it with the graph here. Okay, so basically it's 2 parts of this process. One is then alignment that comes into picture, and second part is basically the physico-chemical properties. Now based on the physico-chemical properties, all cannot be taken. Those ones which have based on the correlation matrix which are orthogonal to each other, that means they are not correlating with each other, those properties could be taken as descriptor values. When you come to the peptide alignment, you can take the most active peptide and align the rest of them against it using certain softwares like Cluster W, and then you can this is how, and you can use equations, and the equations you can use similarity indices, which is used in this case to compare what is the similarity between, in this case, like for example at position like this is position after alignments. Okay, say for example, here this is the peptide here, and aligned with isoleucine has been aligned with aspargine. Okay, the similarity index for that would turn up to be 0.24 for a particular property. Okay, so property is used as a parameter for comparison. So as we have seen here, there are almost 119 properties were taken, but were boiled down to only 43 based on their orthogonal relationships. And then further, a model can be developed indicating this similarity indices and corresponding them to the biological activity. That is basically the HOMOSAR models. We have an HOMOSAR model here with here. So it basically talks about the similarity index, the model here. So it talks about this. This is basically the peptide AB, and that is the reference and the query peptide, and this is the property HS, hydrophobic scale, as in this case. And what is the index? So that would be the value coming up here. So, and again, it looks like it has a negative contribution. So similarity should be a bit diverse at that particular position. So these are the other data sets that were basically compiled in two papers. Basically, the data sets that were studied was angiotensin converting enzyme inhibitory peptides, camel S antibacterial peptides, human amphiphyssin 1 SH3 domain binding peptide. So based on these peptides, we have developed these models individually to validate the method. So this is basically the statistical data for all these three. Try to get as much statistical data as possible so as to validate the model. Excellent. Now, only drawback is that we do not have any experimental facilities, so we couldn't actually synthesize the predicted peptide and test it. So that's one drawback that has been there throughout the research. Next is what I talked about is ensemble QSAR. Ensemble QSAR looks at multiple conformations. The dogma basically is one chemical, one structure, one parameter, which has been a standard standard dogma in the world of binding site analysis. But it is seen that it's not one conformation that binds; it's multiple conformations that binds at time. Only one of them is active. So on basis of that, what we basically try to do is use molecular dynamic simulation. Now this is a tool that is used for carrying out long-term it calculates basically the Newton's equation of motion for small atoms and generates the different shapes the structure can take over a period of time. After that, these conformations, like here as we see here, for this particular pentapeptide, we have generated the conformation. So you can see that there are multiple conformations and all of them switching positions. Okay. Now, using this conformation, how a conformation is defined in this methodology is called as the physicochemical distance matrix. So now in this matrix, basically, this is that physicochemical distance matrix. We look at three values for a particular amino acid. So if this is a peptide, that is VVAC, that is valine, glutamic acid, tryptophan, alanine, and lysine. So if we draw a 5x5 matrix, then the diagonal here looks at the physicochemical properties of that particular amino acid, and these off diagonals basically are the distances calculated. So now the upper bound, basically here, this one calculates the ratio of the alpha distance with the side chain, and here it again calculates the alpha distance with the alpha distance. That means, okay, so if I explain it on this particular graph, the distance of this atom, alpha carbon, with the side chain, okay, are depicted in this quadrant here, on this off diagonals, and the distance between alpha to alpha is depicted in this particular region. So that's how, so it will be, now since off diagonals it will be zeros, we have tried to fill it up with the distance matrix, so physical properties, and therefore we call it as the physicochemical distance matrices. And so, like for example, the distance of alpha carbon of valine to the alpha carbon of glutamic acid is 4 nanometers, okay, and 4 angstroms, and the distance between valine carbon atom to the the alpha carbon of glutamic acid to the side chain last carbon, last atom of the valine residue, or the centroid in this case would be 0.9 angstroms. Okay, so this way we calculate the matrix, and then this matrix is converted into eigen values and eigen vectors, and then we use the eigen values in converting to explain the equation, and that's how we get the ensemble QSR model. Now the statistical tool that has been used in all of these methods is actually a combination of genetic function algorithm and the partial least squares, commonly referred to as GPLS. So with this method, you have tried to propose certain models that can be validated and incorporated into certain software programs for peptide designs. So these are the methods. That's what we look forward towards collaboration wherein these can be interacted out. So with this, I would wind up my topic on peptide QSAR. Thanks to University Grants Commission, who has made it possible for me to reach China. They have helped me out with the travel grant. Professor Evans Coutinho at Bombay College of Pharmacy, Mumbai, India. He is my mentor for PhD. And these are my two angels who are waiting back for me eagerly at home. My contact details are here. And finally, thank you.
Do we have time to take questions?